Swayam Prabha. Digital India. Educated India.
Students, welcome back to Developmental Biology course again. Today we are going to have a new topic, and this is going to be what we call as Evolutionary Developmental Biology. So, I am sure by now all of you have heard the term evolution. Okay, so the theory of evolution is basically the unifying central concept of biology. So as famously told by Dobzhansky long time ago, nothing in biology makes sense except in the light of evolution. So the process of evolution operates by natural selection of the fittest. So this is what you would have learned from your high school and in earlier classes in the college or university. So now for the natural selection to select the fittest, you need to have variations in the population, in a given population. For example, let us say in a human population, if everybody, let us say, is uniformly 5.5 feet height, then there is no way to select based on height, because everyone will be equally competent if height becomes desirable. Let us say shorter height is desirable for a given environment. So variations have to exist in the population, then only you can find the shortest person who might fit a given environment. So therefore the basis for natural selection to work is the availability of variations in a given population. So how are these variations created? Where do they arise from? So they have to arise from variations in the development. Okay. So variations in development is what is going to result in a population of human beings in our example with varying heights. So how are these variations permitted in development? So what are the mechanistic basis of these variations? And understanding of that is really at the center of understanding the mechanisms of how evolution operates. And in addition, what are the variations that are permitted by a given the developmental framework that exists. Like for example, if Hox genes that we discussed in the last class determine the anterior posterior pattern and segment identity, for example in our vertebral column, now what variations are possible within that, and what are the limitations and constraints, and that is going to determine what is available for evolution to work on. And therefore understanding the developmental mechanisms of these features are really critical to explain what is possible in evolution and to predict what kind of structures are going to happen and how that is going to be adapted to a given environment. So in this sense, evolution and development are very, very tightly related, and people very affectionately call it as Evo-Devo. Okay, so that is what we are calling as evolutionary developmental biology. So this is what we are going to discuss in today's class. So as I just said, how are the variations generated? If so this is not an easy question, it is quite complex. So consider the following points and you will understand why this is not straightforward question. So we learned, I will take the example of the body patterning in the Drosophila or the mammalian axis formation that we discussed in the last class. everything seems to be very fine tuned. You know, if you want the chicken type of cervical vertebral column, then you have to have Hox gene stopping a little earlier than, you know, a little, it should restrict a little bit to the posterior compared to what it is in mouse. Okay, so these seems to be very fine tuned and very delicately balanced and tightly controlled. If that is what is the basis of development, then any variation in that could end up destroying the whole organism, right? The structures will just not fit with the rest of the body plan, and as a result that embryo might die. So, how variations are permitted in such a complex and fine tuned developmental program? So, that is one question. Second, most of us from our molecular biology classes, we would have already learned that DNA replication is error prone, the repair mechanism and proofreading notwithstanding, and as a result you do get mutant versions of proteins. And if these mutant versions can have differing activities and that would cause variations, then these proteins will be expressed everywhere. Okay, we discussed at length about the genome equivalence, like all our somatic cells more or less have the same genomic content. So, that means the mutant protein will be there in all parts of the body. Then how that is going to help us create variations? Okay, with like for example, if a protein is involved in height and a mutant version of that protein is going to increase or decrease the height, will that protein not have impact in the rest of the embryo development other than height? Okay. So given the advent of molecular biology does not provide a straightforward answer to this question of how are variations generated. Okay. So, the answers actually come from developmental mechanisms, people who studied development in multiple organisms, and remember in very early classes we have learnt about comparative anatomy, and even in Hox genes we saw the comparative development, like comparing the expression pattern of Hox genes in chick versus a mouse and the number of cervical vertebrae in chick versus mouse, right? The comparative developmental biology, so we already are familiar with, okay, and that is what provides answers to this question of how variations arise. And the answers are quite simple in two phrases here summarized. One is modularity, we will see what that is, and another one is molecular parsimony. Okay. So, these two we will discuss at length to understand how these two features that the developmental biologists have learned provide answer to this question of how variations arise. Okay. So, modularity, we are somewhat familiar with this term already when we discussed about enhancers. Okay. So, enhancers are the ones that modulate the transcriptional rate of genes. Okay. These enhancers can be upstream in the promoter region, or it could be anywhere else, even in the intron or coding sequence, and these are cis acting elements, DNA sequences that modulate the rate of transcription. Okay. And we saw that these exist in modules, like for example in PAX6 gene you have an enhancer that works in lens, another enhancer that works in pancreas, and so on. So, each module is active in one tissue or one time point and not at the other another tissue or time point. Okay. So, therefore, they are modules. You have a lens specific module, you have a pancreas specific module, and so on. Okay. So, these are modules of DNA sequences, and similarly there are developmental modules also exist. Okay. So, these are discrete and interacting modules, anatomical, okay, so that's what I am talking about, to distinguish from the enhancers that you are familiar with. So when you think of modules, do not always think enhancers even in this narrow context. There are other modules like tissue modules that exist. Examples are morphogenetic field, okay, so you have the retinoic acid posterior to anterior, a morphogenetic field, or you have FGF gradient from posterior to anterior, and these gradients act as modules because they are independent and they function completely independent of the rest of the embryo, okay. So like for example, if you take our epithelial mesenchymal interaction discussion, where we saw a mesenchyme from a certain region induces the epithelium to form corresponding structure, be it in birds, the body feather, or it could be the scales in the thigh region and claws at the tip of the limb. Okay, so these are all modifications of the epithelium, but they are instructed to make the thigh feather or the body part, the wing feather or the claw or the scales by the underlying mesenchyme. Now if you mismatch them, what you find is the epithelium acts as a module. It listens to the underlying mesenchyme and then it behaves that way, okay. So we saw the same in embryonic transplantation experiment between salamanders and frog. So salamander makes the structure it normally makes based on the modular instruction coming from the frog underlying tissue that was transplanted, okay. So these are modules, so they function in a way that is independent of what is happening in the rest of the embryo. Some of the cell lineages and the insect parasegments, they all do that. So we saw antennapedia not being expressed in the posterior most thoracic segment meant it did not make wings. But if you do not have the posterior Hox gene, that ultra pythorax, now antennapedia extends into the third segment, and now the third segment behaves like the second segment making a wing. So these are all acting like modules, okay. So these modules therefore, depending on where they are, they could actually modify the structures. So we will see many examples where it will all become very clear. The modular behavior of tissues and the modular behavior of DNA sequences that regulate transcription by stretching them in terms of spatial expression pattern or temporal expression pattern, you can modify the structures, say converting a normal fingers like digits into webbed structure making the bat wing and so on, okay. So we will see individual examples where it will all become abundantly clear. So the main crux of this is modular units allow certain parts of the body to change, that is why they are independent without interfering with the functions of the other parts. So this is how variations are made possible, where the the enhancers, how a tactful use of enhancers led to variations which led to which became available for the natural selection to work and select what fits a given environment. So we have a receptor on our RBCs and also in many other cells called Duffy. It is a glycoprotein and it is essential for many signalings, and it is an essential protein. And unfortunately, this Plasmodium vivax, a relative of Plasmodium falciparum that causes malaria, this one infects by binding to Duffy protein present on RBCs. Okay. So, many pathogens get into our cells by binding to cell surface receptors. Okay. Many viruses also do that. So, the Plasmodium vivax binds to Duffy on RBCs, and that is how it starts the infection. So, now it turns out that in certain population of human beings in Africa, so they are resistant to Plasmodium vivax, and that is because their blood cells do not express Duffy. So, how come blood cells do not express Duffy and they are still alive? Duffy, I told you, is an essential protein and it is required for survival, and if these people have a mutation in Duffy and it is not functional, then how are they surviving? So, the answer comes from an RBC specific enhancer, not RBC specific because RBC does not have DNA to transcribe from. So, the precursors of the blood cells, okay, in the precursors of blood cell, the enhancer of Duffy gene is inactive. As a result, Duffy is not expressed in the blood cells, but it is expressed elsewhere where its function is essential. So, as a result, this enhancer mutant that specifically turns off Duffy in the blood cells has been selected due to the evolutionary pressure of Plasmodium vivax infection. Okay. So, this is how the modularity here in terms of the gene expression helped in selecting, in generating a variation on which natural selection could work. Okay. So, this is one example. Another example we are going to look at is the a fish variety called the sticklebacks, because they have these structures, spine structures, and therefore they are called sticklebacks. On their back you have these structures, and these are originally present in they were present in marine. They continue to exist in marine, but then later they adapted to live in freshwater lakes as well. Okay. So, these have these really thorny spines at the pelvic region, in the ventral region, sort of posterior of the body, and these pelvic spines help them evade predators because these thorns prick them and they do not like to eat these fishes. So, this is a survival adaptation that helps it to dodge the predator. But when it came to freshwater, there are no such predators for it, and therefore this pelvic spine is not required. But then why should it lose? The freshwater sticklebacks do not have pelvic spines. So, why did they lose? Because there are invertebrate predators here that readily hold these fishes using the spine structures. So, therefore, the sticklebacks that lost the ability to make the pelvic spine got selected in the freshwater. So, now how come this could have pelvic spine and this lost it? So, the scientists tried to do mutations. I will not go into the details because it is a significantly involved series of experiments. They finally, through those experiments, identified that a mutation in a gene called PITX1. Okay. So, this gene is responsible. So, that is what they found. But then when they looked at the expression part, when they looked at the amino acid sequence, there was absolutely no difference at all between the PITX1 protein sequenced from the marine sticklebacks and the freshwater sticklebacks. They were identical. But then when they looked at the expression pattern, what they realized is in the ventral part, this is the ventral view at the bottom, like the belly side, near the pelvic region you have strong PATX1 expression in the marine water fishes, but not in the freshwater fishes, sticklebacks. Okay. And very interestingly, they were able to show that if you take the when you look at this sequence, and when they took an upstream sequence containing all these yellow bars, these are enhancers that that was identified later, but when they took this entire region and fused it with GFP and a reporter protein and introduced into the freshwater fishes, they could see GFP fluorescence in this region of these fishes. And more dramatically, when they used these elements taken from the marine sticklebacks and expressed the freshwater PATX1 gene under the control of these sequences in the freshwater sticklebacks, they developed pelvic spines like this, indicating that and then they were able to find out that this particular region, which they named as hind limb enhancer, had mutations. Okay, the sequence difference between these two varieties were not here, okay, in the coding sequence. Instead, it was in an enhancer, and due to this mutation, this gene is not expressed in the pelvic region in the freshwater sticklebacks. So here again you are seeing another example of how turning off a certain enhancer module helped in turning off that gene expression specifically in one part of the body, but not in the rest of the places. Okay. So these are two really good examples explaining the role of modular enhancers in generating developmental variations. Okay. So now we move to the next concept. Okay, so we learnt about what is modularity, but essentially I showed evidence specifically for enhancer modules, and this I kind of indicated some of the things that we have already learnt. Okay, so now we will look at molecular parsimony. What is molecular parsimony and how that helps in generating variations. So parsimony means being miserly, using very few things for doing whole lot of things, that is parsimony. Being frugal, that is what is parsimony. So in developmental biology we call that a small toolkit. So here the developmental mechanism seem to use very few tools to build varieties of structures. Okay, variety of morphology, variety of functions, all generated using a couple of screwdrivers and maybe couple of wrenches, and that's it. No complex tools to build complex instruments. You can easily imagine this in a machine making world, like this screwdriver to tighten a screw in your bicycle can also be used to tighten a screw in an aeroplane. Okay. So you see that we are using fewer tools, but on very complex machines. A similar thing happens in development as well, and that is what we call as molecular parsimony. Okay. So we already have a very good outstanding example in our recent discussions, the Hox genes. In the last class you saw the Drosophila anterior posterior axis, how anterior to posterior different segments, whatever Hox genes determine those segmental identity, the same Hox genes determine similar anterior posterior segment identity in vertebrates. Okay. So Hox gene here being the screwdriver equivalent, okay, only one tool, but building body plan of an insect versus your body plan, an insect is no comparison to you, but Hox genes do both. Okay, so that explains what is molecular parsimony in a nutshell. So this is explained here in some detail. Development differs enormously, but remarkably similar molecules are used in distant phyla. Okay. So, we will see more examples of this as we go along. Transcription factors such as PAX, I told you about PAX working in eye development, lens module I just mentioned, and Hox genes are conserved from cnidarians. Cnidarians are like very ancient organisms like sea anemone and so on. So, these are radially symmetrical organisms that formed even before platforms, roundworms and insects, etc. Just little bit more sophisticated than sponges, but no more than that. So, starting from cnidarians to primates, that is us, these genes do similar things. Okay, their sequence is conserved and you are going to see that they do very similar functions as well. In some instance, one example is right here. PAC6 specifies light sensing organs in mollusks, okay, like shells, clamps and so on, like the octopus. So, they all are the mollusks, and some of them have very rudimentary eyes and some have, like for example, octopus has a lensed eye like us. So, there again PAC6 does that eye development job, and it does the same in us, okay, and in insects. So that's a use of PAC6 over a period of about 700 million years for doing the same job. It is not that sequence alone is conserved, the function, the very specific organ for which it is required is again conserved. And similarly, you have Hox genes. We have talked enough of it already, but we need to see some more details of Hox genes. We need to get to their evolutionary history. When did it begin first? They specify this anterior posterior axis all the way once again from cnidarians to human beings. Okay. The orthodonticals and empty spiracles that we talked about during the discussion of Hox genes, the very extreme end of the anterior portion, like the head region, I told in the last class that the forebrain and midbrain are determined not by Hox genes but by these transcription factors OTX, and orthodontical ortholog in vertebrates is what is OTX, and this gene is required for the extreme head development both in Drosophila like invertebrate and in vertebrates. Okay. So, here is an example when mouse PAC6, I told you PAC6 is required for eye development all the way from mollusks to primates and the insects in between. So, this is an insect example. Here what has been done is PAC6, mouse PAC6 is expressed not in place of where the eye will develop in insect, instead in its leg, but even there it can instruct that leg imaginal disc, the larval organ from rudiment from which or the primordium from which the leg is going to form, that is called a leg imaginal disc in fly larva. And there when you express PAC6, it produces eye in the place of leg. Okay, so that is the level of conservation you see. So, this is called deep homology, which we will discuss in detail a little later. So, here we are seeing these things as examples of using small toolkit. Okay, so starting from ancestral molecules, let us say you have an ancestral Hox and probably that was duplicated and then further duplicated, and two groups of duplicates diverged in different way, forming the anterior group, that is the Antennapedia cluster, and then you have the posterior bithorax cluster. Okay, so that is how it should have happened, and that process we call as duplication and divergence. So, if a particular gene is essential, for early embryonic development, then mutation in that particular gene is going to be lethal. So such things are not going to be available for the natural selection to work upon. But instead, during DNA replication, if slippages happen, you would have learned in molecular biology, and it will be too much for a developmental biology course to deal with those mechanisms. But just take it for granted that there are errors in replication that end up duplicating certain portions of the chromosome. Sometimes it is one short stretch of DNA, sometimes it can be a large part of a chromosome. So these duplications once happen, now instead of one copy of the gene, let us say it duplicated only once and you have two copies. Now the evolutionary pressure is sort of relaxed. As long as one copy remains intact, the embryonic development is going to be alright, okay, and the other copies can be now played with. You can tinker its sequences without having bigger consequence on the embryonic development, and that is how divergence starts, okay. And this divergence now can lead to new functions, new spatially distinct expression, like for example, one duplicate expresses in one tissue and the other one expresses in other tissue, where a slight modification in coding sequence could make that tissue develop differently, and that could become a selection advantage, and so on. So, these duplications enable divergence possible, okay. So many gene families come from such founding members, and then you have a group of related genes. That is how you have Hox gene family, globin gene family, collagen gene family, and distellis family. This is the Drosophila gene. All these gene families arise from, okay, and some of them many encode transcription factors, some are structural, some are paracrine factors, and so on, okay. And so one good example is Hox gene once again, and they come from a single common ancestor. So we do not even know that the earliest original one, but in Nedarians we already have two of them, and so we will see that in a minute. Once I just told you, once duplicated, gene copies can diverge by random mutations in coding sequence, which will be okay, because there is one copy or another set of copies where wild type sequence is present, and it can also be in enhancers, like we just saw PITX1 hind limb enhancer mutated in the freshwater sticklebacks. So due to this, you could get new functions or different expression pattern. Once again, if I go back to that PITX1 example in stickleback, you see new expression pattern. It is not expressed in the hind limb primordium. Okay, so these all become possible once you have multiple copies, and therefore divergence in terms of new functionality as well as new domains of expression both become possible. And such duplicates of same gene with subtle variations present in a given species is called, they are all together called paralogs, okay. So for example, Antennapedia is a paralog of abdominal A, okay, because both are related proteins, they are homeotic genes, and they are more closely related to each other than to other gene families, okay, but they are present in Drosophila itself, and that you call as paralog. But if you take a Hox 4 and of vertebrate, like say human, and look at it in Drosophila, you will sure find it to be more related to Antennapedia to any other gene family, and that you call as orthologs. So related genes present in different species, you call them orthologs. These terms apply to proteins as well, okay. So members of a gene family in a species you call paralog, and members of gene family present in different species, the homology is across species, you call them orthologs. So we continue on that theme to understand some of these dramatic variations that have been accommodated during evolution, and they are presented here in bullet forms, each one different example. So you have different expression and function. If you take distalless, there is only one copy in insects and it is expressed in most part of its body. But on the other hand, if you take in vertebrates where you have multiple copies, for example distalless 3 is required for epidermal differentiation and that is where it is expressed, not everywhere. And distalless 5 is expressed in neural crest only, not everywhere. So there is specialization here, okay. Instead of one person doing all the jobs, now you have employed one more, so then each one can do 2 different jobs to make the whole thing happen. So that is the advantage of this duplication and divergence, okay. And you can imagine from this, if variations in neural crest is going to generate a new body structure that is going to be evolutionarily advantageous, then mutations in distalless 5 could enable that without compromising the epidermal differentiation, because a different paralog is doing that, taking care of that job, okay. Sometimes homologies run very deep. We already saw in PAC6 context in eye development by introducing mouse PAC6 in Drosophila leg imaginal disc, and you have many more such examples. For example, vertebrate HOX6B4 can substitute its ortholog in flies, that is the deformed, and deformed deficient fly, mutant fly could be rescued to wild type by expressing the vertebrate HOX B4, okay. And where it all starts from cnidarians, where you have so again cnidarians are like sea anemone kind of organisms, okay. So these cnidarians they are mostly marine, although some are there in freshwater as well. So they have 2 HOX genes, okay, just 2 HOX, and it turns out one of them that is expressed in the anterior part of its larval stage corresponds to the vertebrate's anterior HOX group, and its posterior one corresponds to the posterior class of HOX genes. So HOX genes seem to have evolved only once before cnidarians and duplicated in cnidarians, then after that the anterior and posterior seem to have duplicated further and diverged to have 5 anterior and 3 posterior, for example in insects, and then they duplicated again 4 times to have 4 clusters in vertebrates, okay, which we saw in the last class. And then so there seem to have been 2 large such duplications in HOX gene families, one in the vertebrates and another separately in fishes. So we are not going to get into the details, but the main point here we want to remember is this. Some of them, like for example HOX4, you have 4 paralogs and all of them do very similar thing, and therefore mutating any one of them is not going to create a phenotype and you need to lose all of them. On the other hand, genes like like HOX11, this one like for example A and D are not expressed in the same place and doing the same functions, which is in contrast to all the HOX4, HOX A4, B4, C4, D4, okay. So they are all expressed same place doing the same things. Here HOXA11 functions both in limb and female reproductive tract, while the D paralog functions only in the mammalian limb bud. So you have both variations, where all retain the same, and in another set where you have divergence of the expression pattern and function. So, in summary, what did we learn about this duplication and divergence? So, these duplications allow redundant structures, like in this case redundant gene sequences, and that allows divergence, okay, because when you have one wild type copy taking care of the required function, then the other copies can be mutated and experimented with, okay. So, therefore, you could have new roles coming up, and that is how these gene families arose. You know, these are just 3 examples. So, you have Hox genes coming from one ancestral founding member, generating a family, but conserved to pattern the body and limb axis, but differently in different organisms, okay. So, the way immediately, let us say hypothetically, immediately after duplication, they started functioning in the body plan, okay, anterior posterior axis and then the limb development and so on. And now the two groups duplicated and made multiple copies, and therefore this body anterior posterior body plan could be altered. Okay, so therefore the body plan of a mouse is not the same as Drosophila. Okay, so but still they have the conserved role in the body plan. Okay, generally speaking, body plan, yes, Hox genes are required for body plan, but the variations in them, like among their paralogs, creates the variations in the body plan that you see between the two groups of organisms. And similar is true with the distal genes, distalless. So, they are required to extend appendages and to pattern vertebrate skull, and variations in them in the paralogs again creates variations in the appendage and the skull structure. And similarly for muscle development, myod family members and so on. So, therefore, the main point here what we are realizing is the difference between the flies and humans do not arise by having different genes, but by deciding which gene will be expressed where and when, okay. So, it is not the humans have new genes, but that where flies only have one, humans have multi-gene families, okay, and that is how the difference between the two organisms arise. So, now let us look at what is deep homology. I kind of alluded to this earlier on when I discussed about the PAC6 and the Hox at the beginning of molecular parsimony discussion. So, let us look at this deep homology. So, deep homology is, you already know paralogs are related, orthologs are related, that is at an individual gene level, and you say they are evolutionarily conserved. But there are situations where the multiple genes functioning in a pathway, let us say signal transduction pathway, they are all conserved and they do the same function as well. When you see such things, you call that as deep homology. Okay, so deep homology, you do not use that phrase to explain how two genes are highly conserved, okay. So, high degree of conservation is not synonymous with deep, the depth of homology. When you say depth of homology, what you mean is, it is not that the individual gene is conserved, it need not be highly conserved. So, it is actually a set of genes functioning in a particular process, let us say limb development. All genes involved in that, they are all conserved and do the same thing, and that is when you call as deep homology. An outstanding example is what is shown here, the way the short gastrulation of Drosophila gene and its TGF beta pathway ortholog decapentaplegic DPP function. So, this is really ancient organism compared to mouse shown on the right. Okay. So, if you look at the fly, so the DPP it determines the dorsal part. If you recall our discussion on the dorsoventral axis formation in the Drosophila oocyte, so we learned that the dorsal protein enters the ventral nuclei at the syncytial stage, and as a result dorsal is expressed only in the ventral, and the dorsal inhibits decapentaplegic expression in the ventral side. So, DPP is required therefore for countering the activity of the ventralization signals, and as a result it allows the central nervous system gene expression in a certain order on the dorsal side. Okay. So, that is what, so these are the genes that need to be expressed, which these are induced by the short gastrulation, and they are restricted to this portion, the ventral side, because of the dorsal expression of DPP. So, this is how these are all central nervous system genes. So, this is the mid body point here, and this requires the highest expression of SOG, and as it progressively decreases you have these genes activated, and these are countered by the DPP expression. And very similar thing, but an inverted structure is what happens in the mouse. So, the central nervous system, for example in our body, also in mouse vertebrates, is on the dorsal, like at our back. Okay. But on the other hand, if you go to invertebrates like C. elegans or Drosophila, it is on the ventral side of the body, and that is because of this expression pattern of these genes. But the main point is not the dorsal or ventral. The main point in our current discussion is the central nervous system formation. That is done by SOG DPP opposite gradient in Drosophila, and that is true here again. It is just that the dorsal ventral are inverted, but the chordin strong expression is what determines this expression pattern, and BMP4, the DPP homolog, determines here this pattern. So, it is just this inverted here. So, this sort of a thing, same genes doing the same thing, this chordin BMP4 or SOG DPP setting up the expression pattern of genes involved in central nervous system specification is the same in both, and this is an example of deep homology. And this also tells you that the central nervous system did not evolve multiple times independently. Then you will see multiple ways of doing it. Here what you are seeing is the same DPP SOG or its equivalent chordin BMP4 is the one doing the CNS specification in both these organisms. And not just that, this is how even the cnidarians seem to do. And therefore, in the entire animal kingdom, how to make central nervous system seem to have arisen only once, and then we have diversification from that. And this is what you call as a deep homology. Okay. And the next one we are going to discuss are what we call as the mechanisms of how all of this happens. So, think of some of these evolutionarily novel structures, okay, that did not exist in the organisms that existed before a certain group. For example, neural crust or teeth, teeth, think of sponge, there is no teeth, teeth had to come as a novel structure. So, how do you make these novel structures? By having small changes in protein sequence or even in enhancer module. How do you make these novel structures? And another major worry comes when you look at the sequences of human and chimpanzees. Okay. So, the genome is nearly identical, okay, more than 99% identical, and many of the alleles of proteins again are very similar. Okay. And but the reality is chimpanzee is not human, it can't teach an NPTEL course, only human being can do. So, where is this big difference between chimpanzees and humans come from? And that is not, that key difference is not in the alleles of protein coding genes. Instead, it is in the sequences of sequences that decide where, when and how much the genes are activated. Okay. Will it come early on, and will it be there in the limb bud, or will it be in the imaginal disc, and for how long will it be expressed there? Okay. So, we saw one example very early on when we were talking about von Bayer's principle and Darwin's descent with modification. So, we saw that the duration of FGF expression can determine whether a dog will have a long leg or a shorter leg, right. For how long the proliferation of those chondrocytes will happen before they enter into differentiation. Premature entry means short leg, they differentiate into bone very early without producing lot of precursor cells. So, changing the duration can also alter the structures. So, these sort of where, when and how much is what really makes the difference between chimpanzees and humans, not the exact protein or the sequence itself. Okay. And how do you make these changes? How they are classified into 4 groups: heterotopy and heterochrony, heterometry and heterotypy. Okay. So, all of them will become abundantly clearer and when we look at individual examples. So, heterotopy means different location. Okay. So, for example, if a particular gene is expressed in one particular location so far in the course of evolution, now the gene expressing in a different part of the embryo leading to a new structure development. Okay. So, that you call as heterotopy. And heterochrony is changing in time. Okay. This changing in time could be not simply the gene expression, this could also mean the rate of development. For example, quicker proliferation of cells versus slow proliferation, or proliferation for longer time than what it happens in another organism. So, those kind of time dependent variation of developmental process or gene expression, both of them we call as chrony. Topy is spatial changes in spatial expression, chrony is changes in temporal expression. Okay. So, in the next class we will get into examples of these, and we will try to understand how these variations are made possible and how it all ultimately explains how variations in development are generated. Okay. So, we will take up the change in location examples in the next class.